Good morning, everyone. Now it's my great honor to introduce Dr. Louise Chow, Professor Emerita of Biochemistry and Molecular Genetics at the University of Alabama at Birmingham. Professor Chow's career brings together molecular genetics, virology, and electron microscopy. In 1977, at Cold Spring Harbor Laboratory, she used the electron microscope to visualize human adenovirus DNA-RNA heteroduplexes, which led directly to the discovery of messenger RNA splicing and alternative RNA processing. That collaborative discovery became a landmark in modern molecular biology and formed a critical part of the work later recognized by the 1993 Nobel Prize in physiology or medicine to her collaborator. Over 4 decades, Professor Chow has investigated the pathobiology of the prevalent human papillomavirus, namely HPVs. Her discoveries significantly advanced our understanding of how HPV gene expression, DNA amplification, and host cell interaction pathways contribute to infection and cancer. Professor Chow's honor includes election as an international member of the U.S. National Academy of Sciences, election to Academia Sinica, fellowship in the American Academy of Microbiology, the UAB President's Medal, the American College of Physicians Medal, the National Taiwan University President's Award for Distinguished Alumni in Science, and Anderson Family Endowment Chair. Please join me in welcoming Professor Louise Chow.
Thank you. Thank you for coming, and I want to thank the generous introduction. I'm very pleased to have this opportunity. Today, I'm going to only talk about the history of the discovery of RNA splicing, and have no time for the bulk of my research career, which is papillomavirus. But here we go. I was born in Hunan, China. I grew up in Taiwan and graduated from National Taiwan University in agricultural chemistry. Then I went to Caltech in the chemistry department. I have no lab skills, no experience, and do not speak any English. So I have to learn it. We learn English, but we don't speak English in Taiwan. So after graduation, I postdoc briefly in UC San Francisco. And then I went back to Caltech in the old lab, Norman Davison's lab, and married Tom Broker. We stayed married. He's here. And he is also a life collaborator in the laboratory in science. So we went to Cold Spring Harbor in '75, stayed there till '84, when we switched to study human papillomavirus because we needed an environment we can collect looking at patient specimens. So I went to University of Rochester, collaborated with the pathologist, and went on from there. And then from '93 to present, I'm at University of Alabama at Birmingham. So at the time, the best tool to analyze sequence relationship between 2 DNA populations is by electron microscopy examination of heteroduplex between the 2 DNA populations. And Norman is a pioneer in this technique. And I learned the art of this particular analysis, and actually the only analysis available to us. I study phage and its host, bacteria. I even studied bacteria ribosomal gene arrangement, so I'm not afraid of a complicated system. So when we moved to Cold Spring Harbor, I initially studied phage mu recombination and helped discover that the inversion in gDNA in one particular segment determined its host range. And that was a big deal then. But also study bacterial insertion sequences that can mobilize drug-resistant gene to other sites, for instance, the mu phage. Then, I want to show you when we first get to Cold Spring Harbor, what we look like. These are the 3 members of the electron microscopy lab. The lab has a very old Siemens we inherited from the previous microscopist, and I don't like it, okay? Very user-unfriendly. So I just walked down the Downtown Road in Cold Spring Harbor lab with my dog, about 1/3 of a mile. We spent hours together in the new microscope room, which is a summer teaching lab. So we used that the rest of the year and use it occasionally in the summer if they're not used. And we just spend hours, keep each other company there in the dark. So the central dogma at the time for gene organization expression is one gene makes one protein, and going through transcription, translation, through the messenger RNA. So there's a bunch of Nobel Prize for all this stuff. But interestingly, discovering a messenger RNA was never awarded the Nobel Prize because it involves many, many people over many years. So they just gave up. So chuck it to history. So it's one gene, one RNA to one protein. But when one investigator moved into higher eukaryotes, and immediately it's obvious in the nucleus RNA is transcribed in the nucleus. It's very heterogeneous in size, but mostly it's very, very long. But in the cytoplasm where messenger RNA is being translated, it's very short, okay? And they are post-transcriptionally modified at the 5' end with a cap, 3' end, the poly-A. So how did this happen? And it's hypothesized the heterogeneous nuclear RNA, larger RNA, is the precursor to the short messenger RNA. What mechanism created short RNA? So the hypothesis, you cut off the 5' end, you cut off the 3' end, or you may cut off both ends. So the model system used to probe eukaryotic gene, biological process is adenovirus. This is a human DNA tumor virus. Even though it doesn't cause tumors in human, it causes tumor in rodent cells. So it has a linear double-stranded genome, about 36,000 base pairs, and it encodes a large number of capsid proteins. So you have a ready source of the protein already. And the question is, where are these genes encoded on the genome? Where is the promoter, and what is the transcription coordinated, and how? So at that time, as I said, there's very little molecular biology, no DNA sequencing is at the infancy, and no reverse transcriptase, and cloning is just starting. So to map these genes, people the investigator using RNA hybridized to restriction DNA fragment of the adenoviral DNA, because a small number of restriction enzymes available, so the mapping is very approximate. And it has, as luck would have it, and in 1976, our old professor, Norman Davison, two other labs, Ron Davis and Dave Hawkins, both are Caltech graduates on the faculty in Stanford. They all discovered that RNA, DNA heteroduplex is more stable in a solution containing a denaturing agent called formamide. So RNA in the right condition can displace the complementary homologous DNA strand, forming a loop, okay? That's called R loop. And the structure can be visualized in a microscope. And those experiments are demonstration and all conducted with one gene, one RNA. And again, Tom and I are not afraid of complicated system. We decided to tackle the entire adenoviral genome to find all the abundant late RNA, where they're coming from, where they're encoded. And so this is one example. You can see, I'm not so sure where can you see? No? Okay. The double-strand DNA is sinuous and smooth, and but you can see 3 loops along the DNA strand. And so 3 genetic blocks can be mapped easily here. So we analyzed many loops and defined the major coding body of these messages. And this paper is published in Cell 11. And that's the first time anybody tackled the complicated system. But there's many unexplained, uninterpretable observations made in adeno-RNA research by Cold Spring Harbor colleagues. Just summarize quickly, many late RNA hybridize weakly to a restriction fragment known not to code the main body. They are all upstream from the main body. So everybody check is just unfortunate, advantageous hybridization, tangling. And the second one is that from Giulianis and Roberts, our colleague, and they studied the 5' capped oligonucleotides and found there's one dominant cap with a 11 long. It's capped at one end, ended with a G. It's created by a nucleus when they generate a capped oligonucleotide. So that's puzzling because there's so many messages why there's only one major cap. So either all of them have the same cap or only one has a cap, the others are not capped. So a graduate student, Dan Classic, just purified 2 late RNAs and found they have the same cap, but this cap is not protected after hybridization to the coding region after RNase digestion. So that's odd. Why is it not protected? Then Roberts had a staff and scientist called Saida Zhang. Her job is sequencing a fiber gene. And she couldn't find any sequence, the 5' end sequence, that can encode this capped oligonucleotide. Remember, those are not sequenced, but by composition. You can sequence RNA there. So this evidence, including when we look at EM in making our loops, we find 5' branch, 3' branch at the end of the R loop. 3' you can easily explain as being poly A, but the 5' end, we don't know where it came from. So the evidence point that there are something strange about these 5' ends. Okay, and at that time, there's a hypothesis suggesting eukaryotic RNA transcription is primed by small RNA that's encoded elsewhere, just landed on different gene primed by elongation. And so happens, adenovirus produce a very abundant small RNA called VA-RNA. So it's encoded upstream from all the known late gene coding region. So our colleague Rich Roberts is absolutely convinced that the part of all the VA RNA serves as the mRNA primer and synthesize the message by elongation, show like this. So primer will dock, then elongate, primer will dock, and so on and so forth. So making individual messenger RNA by docking, primer, and elongation. So and he wanted to test this hypothesis. So he proposed to us a collaboration, and he wanted to hybridize an adeno DNA restriction fragment to our preformed late RNA R loops. So we cook up the R loops first. The chosen restriction fragment had the VA coding at very near the end of the restriction fragment, so for easy orientation identification. However, we know from Norman Davison's lab, the RNA DNA, even though it's more stable, but the stability is depending on the length of the heteroduplex as well. So since we don't know how long, it could be the entire VA, could be part of the EA, could be even 11-mer. So I insisted to Roberts and his research scientist, Jelinus, that they have to give me separate strands of the restriction fragment. Otherwise, the two complementary strands will preferentially reanneal, renature, and driven out the potentially very short and stable RNA DNA heteroduplex. So, and we also have to design hybridization conditions to maximize the probability of this pairing. We have no way of knowing how long, how stable. So this is illustrated. We'll perform an RNA loop, okay, our loop. 3' ends sticking out, 5' ends sticking out. And then so then we add a single strand restriction fragment, and that will hybridize nominally to the VA coding region. And then you can see the structure there like this. But on April 3rd, 1977, we remember clearly that the EM result immediately revealed that VA has nothing to do with the genesis of viral late RNA. In fact, RNA priming was not the mechanism. And rather, what we observed is multiple RNA species displayed in our loop will hybridize two and totally unanticipated, not anticipated sites forming a single strand loop on the 5' end of the message. So neither of which correspond to VA. So VA is not involved. And then we have a perfect negative control. We throw in the other strand of the restriction fragment, and we don't see any hybridization, we don't see any loop structure. So it's not an artifact. So we know we're on something, and something big, because this is unheard of. Yeah. The message containing sequences are far away from its coding, gene coding region. So the did extensive EM analysis using a whole series of restriction fragments, separated strands, to map how many we call a leader, because they are not part of the gene, the translated gene. How many leader segments are there? And we decided that at the end, we mapped 3. It's called tripartite leader. And they are all upstream. They mapped the position at 16.6, 29, 19.6, 26.6. So in between, sequences deleted. So the tripartite leader is formed. And by EM, we estimated the length of the total leader is about no more than 200. And 2 years later, one of the late messages was cDNA was cloned and sequenced, and our numbers are right on the dot, 200 bases. So we conclude the various late MRS species was derived from a primary transcript by splicing to remove intervening sequences of different lengths to link the tripartite leader to different coding regions. So this is called alternative splicing now, because you have a choice to hook up the tripartite leader to different genes. So this is like editing a film to give different results. And this is illustrated here. You have a DNA, you make a long primary transcript, and then you have 4 segments, 1, 2, 3, 4, that's to be kept in the message. In between, that's called exon. In between sequences are removed. Those are called introns. So you splice, you make a short, mature RNA. Now, for illustration purposes, I'll show some example, but this time we used single-stranded template DNA, and then we hybridized to bulk RNA, and then you can see the exon is kept in the message, introns are spliced out. So you form 3 loops for 4 segments, okay? 4 segments are conserved in the message. So I'll show you some example later. In 2014, at Cold Spring Harbor, organized by James Darnell Rockefeller, all the attendants are by invitee. All the speakers are invited. And the title of the meeting is History of mRNA. And this title, EM Solved the Problem, was not what I made up for my talk. It's James Darnell gave it to me. Say, this is your title. So indeed, it's absolutely true. EM experiment explained all the puzzling observation by the Cold Spring Harbor lab. And in fact, then classic, the graduate student, now able to protect the cap by hybridizing the messenger RNA to the coding region of the first leader, and showing now it's protected from RNA's digestion after hybridization. So this confirmed our EM mapping, the RNA 5' N and promoter region. So I'm showing you some images. Now, this is a single-stranded DNA hybridized to message. In the top panel, you can see 3 loops, small, medium, back to small, then continue the message body. And the bottom strand, you can see the first 2 loops are the same, but 3rd loop is different size. Okay, now we are using the tripartite leader to access to a different message. And this is a complicated loop structure from one of the rightmost message code fiber. Here we have the same 2 small, medium, a very large group, but we didn't go directly to the message. There's a 4th loop. So this message has 4 introns. So 5 coding regions, basically, with conserved segment. And the bottom one is similar to that, except the 4th intron is tiny, tiny. It's the same message. So the large loop becomes even larger. So I have to, visualize this message, look on the screen and capture in a field and analyze them. So these are all single cell, single molecule analysis. Every day now, people use single cell analysis. In old days, this is all manual. No any program or a tool is to analyze except by hand. And in this same work, we also discover adeNO-RNA from the rightward and the leftward transcription can splice in the middle of coding region. And you can see it from the interpretive diagram. And the RNA is continuous. There's an intron, double-stranded intron looping out. So in Cold Spring Harbor Symposium on chromatin in 1977, June, Tom Broker presented a talk summarizing the EM results and the biochemistry done by our colleagues in this symposium. And the phenomenon of RNA splicing was announced, and the audience is pretty stunned, but they didn't question it. It's immediately accepted. This new process in higher eukaryotes, because the data Tom presented is compelling. And the second reason is that a number of investigators in the audience, they study virus or human or animal genes, have uninterpretable results. Now they say, it's RNA splicing in their system, but they didn't know. They just chuck it away, hidden it away. And so, but Cold Spring Harbor director allowed the symposium participants to incorporate this RNA splicing phenomenon to reinterpret their data when they submit their article for the Cold Spring Harbor publication. So if you look at the volume, there are many papers reporting splicing at the same volume. So at this meeting, we also, Cold Spring Harbor, 4 groups submitted 4 manuscripts to, not more than 4 groups because our EM work is two labs. The 4 papers were submitted to Ben Lewin, the editor of Cell at the time. And they all accepted and published in Cell, and the REM paper is the lead article. So subsequent years, we did a complete, very thorough examination of adenotranscription. And you can see there's a tripartite leader, the little tiny lines there, and joined to different coding regions, and those messages have different 3' end poly-A side. So that's also a new phenomenon. And the left strand, right strand, they cover the entire transcription, cover virtually the entire gene. And one of the message actually have 4 promoters, okay? One major late, and then minor promoters. And and this map pretty much still stands after decades. But this is not just for virus, everybody knows. But in those days, DNA sequencing is still very tedious, very expensive, very slow. So I collaborate a lot of research teams determining structures of many animal and human genes. So just to give you an example, this is a laminin B, human laminin B2 gene. The gene span almost 60,000 base pairs. They have 28 exons. And so this is tedious work by hybridizing cDNA to overlapping genomic segments cloned in different vectors. I just give you a visual impression. Here's another gene. It's a collagen, basement membrane collagen. And you can see 6 loops, so it tells you immediately there are 7 exons in this particular cone. And you can see the loops are huge compared to the exon that's preserved very, very short. So that tells you you can sequence the human genome, figure out what proteins, what genes there are. You have to study mRNA. So now, from that time on, there are a major change in the central dogma for high eukaryotes. So RNA splicing can allow for split gene. Gene can have alternative splice, and that's what explains the huge number. There's more protein encoded than there are genes encoded, or protein produced than gene encoded in human genome. There can be alternative promoters, alternative poly-A, and these together will generate a large number of proteins, or even sometimes different proteins in the same genetic region. A famous example is P16-INK4A and P14R. Both are tumor suppressors, disrupting PRB and P53 tumor suppressor. So anyway, the phenomenon contribute to coordination of gene expression in eukaryotes, and with impact on evolution, development, differentiation, genetic and metabolic disease, cancers, and therapeutic applications. And I'm just in awe from yesterday's talk, how much has advanced since 1977. We are like cave person using stone tools at that time. So, but I'm just so pleased that the science advanced so quickly. So then we moved to medical school and study human papillomavirus. And I will just summarize quickly to say, when we start moving into this field, very, very little is known. Okay, they're 7.9 kb long. They encode 2 oncogenes, E67, but they don't know how they know they inhibit epithelial tissue, and they infect epithelial tissue and only human epithelial tissue. They cannot get enough virus from infected patient specimens, and when they do get virus, they cannot grow them. So nothing is known how they transcribe the gene, how many genes are being transcribed. Are they spliced? Are they alternative spliced? And how are they regulated? How the DNA is amplified to generate in a tissue progeny. Nothing is known. So we had to devise many methods to figure out the whole picture. And so it turns out virus is differentiation dependent in the epithelial cells. So we have to grow skin from primary keratinocytes. And then we have to figure out the gene function by testing each gene in the differentiating epithelium. You can't test them in monolayer proliferating cells. They're not properly expressed. And eventually, we recapitulate the productive system. HPV-18 is a high-risk virus. Why do we spend so much, 45 years doing this? This is a medically important virus causes cervical cancer and other anogenital cancer in both men and women. as well laryngeal papilloma, which is benign but is very deadly. But the key thing is that they cause oropharyngeal cancer in men and women. And how much deaths they cause? 5% of all cancer deaths is attributed to papillomaviruses, human papillomavirus. And there's no therapeutics, okay? It's when you have advanced cancer, you just operate, chemo, that's it. You cannot treat them. There is a very highly effective vaccine. And everybody probably heard of this, Gardasil 9 now, targeting 9 types. But that's prophylactic, it's preventive. Doesn't do anything to existing infection. And the virus can be persistent, and they can be reactivated when the person's immune system is depressed. In old days, it's AIDS patients. Now AIDS patients are getting better, but there's still other conditions like autoimmunes or organ transplant. So this still is serious. Our lab are now using different model systems to test potential therapeutic agents. So I didn't waste the last 45 years, just put it that way. Okay, I'd like to wrap up by, I want to wrap up with one slide that's showing Tom. Many you can recognize him, he still pretty much look the same. And his partner in life in science, and collaborators, students.